Hi everyone, I'm Nadia Sellami, PacBio's Global Lead for Biopharma Applications, and I'll be the host and moderator of today's webinar. I want to welcome everyone to this webinar with today's topic focused on the use of amplification-free gene editing characterization using HiFi sequencing. And I'd like to introduce our speaker, an expert on gene editing characterization with HiFi, who is Dr. Adam Ameur, who is Associate Professor and Senior Bioinformatician at the SciLifeLab National Genomics Infrastructure in Uppsala University in Sweden. In the first part of our webinar today, we'll hear an introduction by Tseng from PacBio with an overview on how HiFi sequencing for gene editing applications can be used. This will then lead us into the talk by Dr. Ameur on how he uses HiFi sequencing in his work and showcasing an amplification-free approach. We have a lot of great material to cover today, and both the introduction and Dr. Ameur's presentation are pre-recorded, but will be followed by a live Q&A session. You're welcome to submit your questions at any point during the webinar by typing them into the area provided on your attendee control panel, and we'll answer as many questions as we can at the end. We've also uploaded complementary literature to your control panel, so please feel free to download this and take it with you. We'll be recording this webinar and making it available for download in the coming weeks, so please keep an eye out for a follow-up email with a link to the recording. And immediately following this webinar, you'll receive a brief questionnaire. Please do take the time to fill it out as it helps us understand your needs and plan for future webinars. And you can stay up to date on upcoming webinars and events by visiting and bookmarking pacb.com/events. So with that, let's get started with the talks before we have the live Q&A.
Good morning, everyone. My name is Elizabeth, and I'm Associate Director at Integrated Marketing at PacBio. Today, I'm going to talk to you about using HiFi long-read sequencing for gene editing research. We have come a long way since the first human genome was completed in 2003. In the 2 decades since, we've seen the development of CRISPR-Cas9 as a gene editing technology. 10 years later, we saw the publication of a truly complete telomere-to-telomere human genome assembly, this time filled in with the gaps of 8% of the genome region that contains many disease genes. Then, a year later, we saw the approval of the first CRISPR-based therapy, Casgevy, in the US for treating sickle cell disease. Gene editing is really one of the most exciting technologies of the next decade. In the past 10 years, the field has been advancing, creating platforms and animal models for gene knockouts, genetic screening, and other ways to edit the genome. In the next decade, we can see a demand for further innovation in therapeutics and agricultural biology. However, despite its great promise, safety remains one of the key challenges for editing-based therapeutics. The common concerns are off-target editing, where sequences outside of the target gene are unintentionally edited, editing errors in the target region that introduces indels and sometimes large structural variations, as well as CRISPR-induced Mosaicism, where the editing gives rise to multiple outcomes. So where does HiFi technology fit into this? HiFi long-read sequencing is capable of sequencing large DNA fragments of up to 25 kilobases with 99.9% accuracy. It can sequence through highly repetitive regions with no sequencing bias. And if sequencing native DNA molecules, such as those from whole genome sequencing or unamplified DNA fragments, we can retain the methylation signals. In fact, the long read length, high accuracy, and preservation of native DNA methylation signals is one of the reasons why HiFi sequencing was used to generate the first truly complete telomere-to-telomere human genome in 2022. This effort filled in the 8% of genome missing from the original reference, including regions containing many disease-relevant genes. While HiFi has many other applications, including producing high-quality plant and animal genomes, full-length bulk and single-cell transcriptome sequencing, for the remainder of this talk, we will focus on what HiFi can do for assessing gene editing outcomes. And now let's talk about HiFi specifically for on and off target editing assessment. Here are a couple of studies that used Amplicon-based sequencing to characterize gene editing outcomes. In the top study, large amplicons of about 5.5 kilobases were sequenced, where researchers found that about 7% of the insertions and deletions were in fact larger than 40 bases, sometimes up to several kilobases. These long indels would not have been able to be observed with short reads. In another study, structural changes and complex rearrangements as a result of editing were observed that were also not identifiable with short reads. In another study, insertions of a construct into a target locus using homologous recombination were characterized using amplicon sequencing. HiFi was able to sequence the construct and the flanking regions to determine the precise integration site. The researchers found a high percentage of integrations were actually imprecise, and the bottom figure shows there were further characterizations of the different types of integration. So we can see that HiFi for amplicon sequencing is able to characterize integrations, structural variations, as well as indels. However, there are limitations with amplicon-based sequencing. In particular, PCR-based methods introduce amplification bias and errors, and they eliminate native DNA methylation signals. As a result, coverage between alleles may be uneven, low-frequency variants can be harder to detect, and it can become difficult to assess whether gene editing has altered methylation in ways that could affect cell function. Let's compare PureTarget to other common targeted sequencing methods. PureTarget sequences unamplified DNA, so there is methylation information retained, no PCR bias or error. Further, it enables you to run a single panel with a pool of dozens of targets. And with its latest product, it is high throughput, where on a single Revio system, you can process up to 100,000 samples in a year with 96 plex per smart cell and automation. Lastly, PureTarget gives you high coverage depth so you can be confident in the results. How does PureTarget work? We start with high molecular weight DNA at as little as 1 microgram. We load the Cas9 enzyme with a panel of guide RNAs that cut on both sides of the targets of interest. After that, we ligate barcoded PacBio SmartBio adapters, perform nucleus digestion to deplete off-target molecules, so you're left with an enriched library of genes of interest. The library is then ready for pooling, cleanup, and sequencing on the Vega or Revio systems. Because of this approach, PureTarget avoids PCR biases, can work for hard-to-amplify regions, including those containing repeat expansion genes, and preserve DNA methylation. This means, in the case of gene editing, one can get allele-specific resolutions as well as all the long indels, concatenators, inversions, and different structural variations. PureTarget is fully supported from panel design using both existing panel genes or custom gene panels, as well as variant calling command line tools. Thank you for hearing about PureTarget for gene editing assessment. And now we will transition to Adam's talk.
My name is Adam Ameur. Today I'm going to talk about how we are using the PureTarget method to study outcomes of CRISPR-Cas9 genome editing. I work at the SciLifeLab National Genomics infrastructure. We are like a big core facility for sequencing, and we take on projects from all around Sweden and also from abroad. We have access, I would say, to most of the sequencing instruments available, and we also try to introduce them and develop new applications. And I have been for a long time interested in long-read sequencing and what we can do with these exciting new technologies. So with PacBio, this is a technology that we've been using already since back in 2013. So you can see the installation of the PacBio RS2 instrument here. It was a huge instrument, but still it didn't have so much throughput, I should say, and also quite a lot of errors in the reads. But this has all changed now, so we installed the PacBio Rebio system in 2023. I think we were one of the first sites in Europe to get this equipment, and now we are trying to use this machine for many different types of applications. And just to mention a few projects that we're working on right now, perhaps the main project that I'm dealing with is human population sequencing. So we have a project called the Genome of Sweden, where we are going to sequence at least 1,000 individuals and hopefully many more. And right now we're also testing PacBio's reusable smart cells in this project. And we were able to generate more than 280 gigabases of HiFi data by using one smart cell 2 times. So that's really exciting, and that can sort of push down cost and make us run even bigger projects. Then I can also mention work that we're doing in single cell sequencing. So we are using long read sequencing to study RNA molecules, which allows us to look at isoforms in single cells, and we're also attempting to expand long read sequencing to look at DNA of single cells. But today I'm going to talk about work that we're doing with long read targeted sequencing, and here I really want to mention my colleague Ida Höjer, who is the one who has been working and developing these protocols that I will talk about today. So she's a very crucial person for this project. And the method that we have been applying is PureTarget. This is a protocol for amplification-free targeted sequencing using the Cas9 enzyme. So basically what it does is to cut out specific regions of interest using the Cas9, and then we ligate these smart bell adapters to the molecules that have been cut. Then in this way, we can enrich for these molecules that contain the target of interest and sequence them on the PacBio Rebio system. And what we have been doing here is to create a custom panel to study the effects of CRISPR-Cas9 genome editing using this method. And the important thing here is that this method is completely PCR-free, so we don't have any amplification, and it also allows us to read methylation information in these molecules. And the question that we want to address is to really understand what happens when using CRISPR-Cas9. I mean, this is a fantastic method for changing the DNA in cells, but there's also been some concern that sometimes the CRISPR-Cas9 system also does other things than what we want. There's also quite a lot of literature, both on papers showing that we don't have any unexpected events, but more recently, sort of you can see some examples here to the right. You can see articles that claim that a lot of strange things can happen in the genome after been doing genome editing. And the kind of unintended genomic consequences that we're interested to study here is first off-target genome editing. So that would be that something happens at some other location in the genome than what we intended. We're also interested in seeing structural variation, so perhaps that portions of the genome can be deleted or inserted at some of these CRISPR-Cas9 sites. And finally, we're also interested in mosaicism. So this would be that different cells contain different edits after have been doing CRISPR-Cas9 genome editing. And the samples that we want to examine is from a project that we ran a couple of years ago in Zebrafish. So we designed a project specifically for studying both intended and unintended consequences of CRISPR-Cas9 in Zebrafish. And the way that this was done was that we injected the CRISPR-Cas9 into fertilized zebrafish eggs. And then from these eggs, we got pools of larvae and also adult individuals. And these adult individuals were then crossed to generate an F1 generation, where we also then have larvae and individuals that are sort of juveniles in this case. And for these samples, we had previously sequenced them using long-range PCR and amplicons of about 5 kbs in size. But now we wanted to sort of revisit these samples with the PureTarget method, and we selected 32 of these samples at different stages in this assay, and we run them on one single SMRT cell. Just a quick slide here on the design of this panel. So we included 7 different targets. So there were in fact 4 different genes that we wanted to edit in this experiment. So we have a gene called LDLRA, one called SH2B3, we have another one called NBL2, and the final one has a very difficult name, but I call it Oaxaca. So we had the on-target sites for these genes, and 2 of the genes also had off-targets, so we included them in the assay. So in total, it was 7 targets in the panel that we created. Okay, so jumping to the result of this experiment. So to the left, you can see the coverage that we obtained for the 7 different target regions and also for these 32 different samples. And a really impressive thing is that we generated over 1000x coverage. So it was 1168x coverage on average for the target regions, which is really great. And to the right, you can see the allele balance for heterozygous F1 zebrafish. So in this case, we expect both alleles to be covered with 50% of the reads, sort of in a perfect world. But as you can see, the long-range PCR generate very biased data. So in some cases, only one of the alleles is covered, whereas the same samples in PureTarget gives very even coverage of both alleles. And here you might ask yourself, why is the long-range PCR more biased? And I think that there are many different reasons. But one example that we found, at least in one of these regions, was a repeat sequence. So in this case, in this figure, we have the PureTarget data here at the top, where you can see both of the alleles. And for allele 2, we have this AT repeat insertion. I think it's about 74 bases, I think, of just AT repeating. And this is not at the genome editing site, so it's in some other region of this PureTarget region. And if you look at the long-range PCR, you can see that almost all of the reads are from allele 1, and then we have just one read in this case from allele 2, and you can see that it terminates within this AT repeat. So this tells me that we have a problem in the long-range PCR. So when it comes to the AT repeat, it actually sort of stalls and ends in there. So we fail to amplify that region. So of course that will give us very biased results in the data. Another advantage with PureTarget, except being very unbiased, is that it also gives very high quality reads. This is one example where we have the Cas9 cleavage site here. It's pictured by this arrow. And you can see in the PureTarget data that it's much cleaner, and you can see exactly which types of edits have been incorporated. It's a 7 base pair insertion on one allele and a 9 base pair deletion on the other. And there are very few errors also in the PureTarget data. And if we trust the quality scores coming off the instrument, then we have a QV39, which would mean that we have one error in about 8,000 base pairs. And since this region now, we have molecules that should be around 5,000 base pairs, this means that many of the reads that we're sequencing will in fact be perfect across the whole sequence. Okay, so that was the data we generated. Now we can look into what types of unintended genomic consequences we discovered. So first, looking at off-target genome editing, I have one example here of an off-target event. This is in the F1 generation. So this has in fact been inherited from the F0 fish, where we have a 3 base pair off-target deletion in this gene called Oaxaca-B, which is very similar to Oaxaca, which was the one that we wanted to edit. But this shows us that, in fact, it's also in a coding part, so it can affect the protein in this gene. Looking at structural variation, we also found examples of such events. So this is a 1053 base pair on-target deletion. This is in the SH2B3 gene, and looking in larvae pools from the F1 generation. So these are 3 different pools of larvae, and we can see that all of them carry this big deletion, at least at some level of frequency. So that, of course, could also be a bit problematic. I mean, unless you wanted to induce this specific event. But what we have been studying mostly in this study is, in fact, mosaicism. And the reason is that we have these very high quality reads and we have so high coverage. So this allows us to look at mosaicism in a very detailed way. This is one example looking at the F0 zebrafish, where we have, again, the Cas9 cleavage site here by this arrow. And if you look at the reads generated here, so this is from one single fish, and remember that the Cas9 genome editing was done in the fertilized egg, so in the single cell stage. But still, this adult fish has many different edits. So you can see, for example, a 17 base pair deletion. We have a 50 base pair insertion. This is a 38 base pair deletion in some other read, a 35 base pair deletion, and so on. So many different types of editing events. And we have been using a software called SIQ to quantify these edits in a more detailed way than looking at just IGV images. So in this way, we can get these types of illustrations. So looking at the SH2B3, we have 3 different fish. And on the on-target site, we had 9 different alleles in one of the fish, and we had 7 other alleles in the fish number 2, and 18 alleles in fish number 3. And similarly, on the Oaxaca gene, we had between 9 and 11 different on-target alleles. So all in all, we found up to 18 different alleles in a single fish. So this tells us that we have a high degree of mosaicism. And what was even more interesting, perhaps, is that we found this mosaicism to be present also in the germ cells of the F1 fish. So I'll try to explain what's going on here. We are starting then from the F0 parents that gave rise, in this case, to 5 different F1 siblings. And looking at the alleles that we found in these individual fish, they have been then denoted by different colors and numbers. And we found we found 7 distinct alleles to be present among these different fish. And if you want to know exactly what these different alleles are, then we also have information from the larvae from the same crossing. So we could see that all of these alleles, in fact, were present also in the larvae. So all in all, this picture shows that we find 7 different alleles in the siblings that come from the same parents. And this can only be explained by the parents being mosaic in the germ cells. Otherwise, we would expect to find at most 4 different edits. And the same story was true also for the other gene here, the Oaxaca gene. In this case, we found 5 different alleles among 4 individual fish. So this, I think, is quite interesting. And you could also wonder then, how does this germ cell mosaicism really arise? And the hypothesis that we have right now is that we're doing the CRISPR-Cas9 injection here in the fertilized egg. So that's in a single cell stage. But what happens is that the CRISPR-Cas9 system keeps being active through several cell divisions. And this process must be going on long enough to make the F0 individuals mosaic in the germ cells. So I don't know enough about sort of biology to understand how many cell divisions these are, but it must continue to be active for quite some time. And as a result of this, the germ cells are mosaic, and we can then observe more than 4 alleles in the F1 siblings. Okay, so just to summarize then the findings from this experiment, we do find off-target genome editing. We find structural variation, and also we can look into mosaicism in a very detailed way, and we find really lots of mosaicism. And all of this can be done very accurately thanks to the accurate, unbiased, and high coverage PureTarget data that we have. And finally, there's also more information in this data. I'm thinking here about the methylation data that we get as an added bonus. So we have 5MC information for every read in these samples. And we did try to look and see if there's any differences in methylation patterns in the edited fish as compared to the unedited controls. But this is looking at two different regions here. So first for SH2B3, we didn't find any differences. So that both the edited and unedited fish are methylated. But for Oaxaca, we didn't find any differences either, but in that case, they were unmethylated. But still, I think this opens up for exciting new experiments, so we can study the effects of genome editing, perhaps in promoters, where we might see differences in methylation too. It could also be used for studying outcomes of methylation genome editing. Okay, so to summarize this study, one thing that was really exciting is that PureTarget gives us these super high quality reads. So it's even higher quality than normal HiFi, just because we have shorter molecules and go around them more times and generate more accurate sequences. We get over 1000x unbiased target coverage, including 5MC methylation. A really important thing is that every read here represents a unique molecule. So basically, there's no need for using UMIs, which is used in other types of protocols. And also, this method allows for multiplexing and can be automated. So we're also now thinking about other targets we can study and also perhaps other types of applications we can use this for. And if you're interested to know more, we have a preprint out on this study. And also, for those of you who are interested to know more about long-read sequencing, I can promote that we are hosting a long-read sequencing meeting in Uppsala this November. So we have lots of great speakers and hope to see some of you there. Finally, this has been a team effort, and the people involved in the project are listed here. And especially, I want to highlight again Ida Höjer, who has been doing a lot of this work. So thank you all for listening, and I'd be happy to take any questions.
Hello and welcome back. So with that, let's go into your questions. I have one question already. The question is, what range of DNA sizes can you put between the two CRISPR sites and sequence?
Okay, so what length of the region is that question? That is a good question, and we haven't fully explored it. But I would think that we can sequence molecules that are 15 kb up to 20 kb. So I think even if we have insertions of, say, 10 kb, we should be able to sequence them. But if we have something like 20 kb, then it might be more difficult. So I think that's about the range.
Great, yeah, and I might add that it might also depend a little bit on the quality of the DNA.
Yeah.
You know, if it's already too fragmented, that might limit the size that you can do. But if you have really high quality DNA, then you have really long fragments.
That's a good point.
Adam, you mentioned that you're considering using the PureTarget method for other applications as well. Can you talk a little bit more about that?
Yeah, I think it's interesting if you're thinking about, finding in applications where you're looking like to find the needle in a haystack kind of thing, where you are traditionally using UMIs. I think this could be a nice way because the UMI it also has some problems actually. So I think if we are able to sequence native molecules instead, that could be really interesting. I think that there could be other types of gene editing applications, but also other fields like cancer, for example. So I think that we will try to explore that in the future.
Great. What editing frequency or percentage do you think might be the lowest that you could detect with this method?
It all has to do with the coverage, I think. Of course, to some extent, perhaps also the quality of the sequences, but I think it should be possible to go below 1%, perhaps 0.1%. If it's possible to go lower than that, I'm not sure. I think the limit might be somewhere around there, at least from a practical perspective, that we need too much DNA otherwise to go lower than that.
Yeah, great. So you've talked about having used Amplicon-based editing characterization first, and then you tried out the PureTarget amplification-free method. Do you see any scenarios where you would still recommend amplification-based methods?
Yeah, I mean, there are advantages of using amplification-based methods still. So I think one is the amount of DNA available. So you can go much lower with amplification-based methods. And also it's a matter of pricing, I guess. I mean, with amplification, you can add barcodes during the PCR, and then you can really drive down the cost. So there are different scenarios, I think.
Great, thank you. I'm also curious, was it previously known that the mosaicism could be inherited after gene editing?
That's a very good question. So I've been trying to sort of look through the literature, and there's not much really clearly stated about this happening. But on the other hand, when talking to people doing genome editing, like the ones who are doing the Zebrafish experiment, it seems to be very well known in their field that they need to keep on doing more crossings because also the fish in this first generation are mosaic. So it seems like there is knowledge about this, but perhaps I haven't found like a single paper stating this clearly.
Yeah, it's really fascinating. Some more questions. Have you ever detected translocation, balanced, dicentric, acentric forms, and were you able to quantify the frequency of the 1 kb deletion event you detected?
Okay, so that's a good question. And the translocations, and this may be one of the weak points of this method, because in this case, we rely on having 2 cuts on the different sites of the edit. So we design the edits to be present on just 1 chromosome. So if we have a translocation, then that will not be seen by this method. So I think that we would need to do some shallow whole genome sequencing to also catch all the translocations and other types of weird kind of events. So we haven't looked into that. And with regards to the frequency, I think that we can get a good estimate of the frequency of at least the 1 kb deletions and things like that, because it's basically just counting the number of molecules. As long as there's no big bias in the length of the molecules being sequenced, I don't think it should be a problem.
Great. The next question, do you think that PureTarget can be used for off-target identification for base editing?
Yeah, I mean, I think that we first need to specify where the regions are. So for that, we probably need other protocols to identify the location. But if we have a known or suspected target or site where we have off-target editing, I think that we could quantify the frequency of the events there as well.
Yeah, great. Do you think it makes sense to use a SMRT cell for a few targets? So how much is the cost for you to run a SMRT cell?
Oh, that's a difficult question.
But maybe
I think that you at PacBio perhaps are more suited to talk to the pricing here.
Right. But maybe let's talk about multiplexing or how many targets you would put on one slide?
Yeah. So yeah, in this case, we just had 7 targets. I think that we could easily do many more. We don't know where the limit is actually right now. Probably 20 targets, maybe 50. I don't know. And I don't think that there's a big limitation also in the number of samples. So I think that we could, put more than 32 samples as we had right now. So in that sense, I think it's might be possible. I mean, if you're doing a large-scale study, sort of looking at the same events through many samples, I think that could be done in a quite cost-efficient manner.
Yeah, and I mean, from PacBio perspective, I also totally agree. Some of our pre-made panels have more than 20 targets, and you can multiplex also, a number of samples with them, and it just depends. It's a matter of the depth that you want to achieve for each of your targets per sample. So, as so many things, the answer I think is it depends, right, on your goals. Yeah. And here's a question on what are the input requirements?
So that I think Ida would be the person who knows more, because I'm more of a computational person. But I think it's around like 1 microgram of DNA, if I don't remember completely.
Yeah, I think so too, and I think you can get away with even less in practice, but that's what we recommend. The next question was about the coverage per target. I think you mentioned that in your talk.
Okay, so what kind of coverage we need? Mm-hmm. This also, I think, depends on sort of the level of safety that you want to sort of look at. And I don't know what will be the requirements for different types of gene editing sort of methods, or maybe it could also depend on sort of what kind of effects it could have, if something goes wrong, and sort of. But I think it's nice to have perhaps 1000x coverage, because then you could go down to 1% quite confidently. So maybe that could be a target, at least for now.
And that's what you saw in your experiment, right?
Yeah, and I think, the important thing is also that this data is quite unbiased. So if you would have 1000 reads from a PCR-based assay, maybe that you don't sort of see all the alleles anyways, but I think having 1000x coverage with these types of reads, I think, would be quite fine to be confident at 1% at least.
Yeah, and you also, so this leads a little bit into the next question, is if you find a length bias for structural variants, for example, would a 2 kb deletion be overrepresented versus the wild type molecule? And is there maybe a sweet spot for length to design the targets?
Yeah, that's a good question. I don't think that we have done all the experiments to know fully about sort of if there are any biases like that. But I think, based on, I think it also has to do with sort of what type of molecules are preferentially loaded on the PacBio SMRT Cell. But if we can get most of the molecules in a range that are so that they're quite similar, then I think it should be fine. But of course, if we have one molecule that is like 10 kb longer than the other ones, there might be a bias, I think. But we haven't studied that in detail.
Yeah, and I may add to that, because I think you're eliminating the amplification bias, which would preferentially amplify shorter fragments, right? There might be slight differences in loading, but originally these PureTarget or pre-made PureTarget panels were designed to characterize and quantify repeat expansions. Yeah. And so we really tried to have a relatively quantitatively accurate representation of both alleles, and to really minimize the size bias.
Mm. Yeah, but I think it's a good point. I think the PCR is the one that's creating the most bias for the length differences.
And then next question, can you please clarify how do you account for the unintended off-target edits elsewhere in the genome? Apparently those will not be cut out by CRISPR during the life.
That's a good question. So in this case, we actually cheated a bit because we had sort of known off targets from our previous study. So we just sort of designed them based on experiments that we had done before. But I think what you can do is either to do like a computational prediction where the off-targets should be identified, or you could do some other type of in vitro assay to first find potential off-targets and then sort of design regions surrounding them.
Yeah. Here's a question that's very technical. I don't know if you can answer this. How much does your yield vary per SMRT Cell?
That's a good question, and it's difficult to answer straight away because we haven't done many of these runs with PureTarget. So actually, this is just from one SMRT Cell. So I don't think we have the data yet to know.
Yeah. So here's a question. I'm not quite sure what the person is asking, but I'm going to relay it to you. Do you integrate the insights offered by this technology to shape your research, or is your goal to verify consistent, successful gene edits?
So I don't know, but my goal is perhaps more to develop the methods to be able to do all of these things. So I'm quite interested in just method development, and then I think it can be used for different purposes. But yeah, and in this case, I think that there's still perhaps a lack of tools that are focusing on sort of detecting these types of unintended consequences of genome editing.
Yeah, and maybe I may add, what I see from our customers is customers also use this, in a production sort of setting to, for example, characterize transgenic animals, and also to characterize, for example, gene therapies and cell therapies where the cells have been modified genetically, like, say, insertion of a CAR construct at a target locus, things like that. So I think it goes both ways on how to use this technology. Another interesting question, how much sequencing yield is lost to the areas outside of the edited regions? And can this lead to confounding results?
I would say that a majority of the reads are lost, actually. So we get a few thousand X on the target site, but I think that we have millions of reads, from the SMRT Cell. We haven't looked at exactly where these other reads are mapping, I should say, yet, but it could be interesting to see if there's anything there, but at this point we've still only focused on the target regions.
Great. And maybe one last question. So you mentioned the SIQ or SIQ software.
Yeah.
Is that publicly available?
Yes, and yeah, I should have put a link. I realized when watching this. So it's our colleagues in Leiden, Netherlands, who are developing the software, and it's published, it's available, and they also have a website. So you can Google it and try to see and find it. It's really great.
So SIQ software, and then people should be able to find it.
I think so, and gene editing, perhaps. Yeah.
Great. Any other future directions or words of wisdom you want to share?
No, I'm quite excited about this. I mean, that we get this number of molecules from a target site and the methylation information. So I think that's maybe one of the future directions that I would like to take, also to dig into the methylation.
Yeah, amazing. Thank you so much.
Yeah.
So I'd like to thank you and all the attendees today, for you, Adam, for sharing your expertise and experience. And as a reminder for everyone, immediately following this webinar, you'll receive a brief questionnaire. So please do take the time to fill it out. And thank you again for joining us today. We hope you'll join us again for a future webinar, and have a nice day. Take care.